curiosity. I think you should never stop asking questions, and you should never stop trying to understand behaviors or phenomena that you see. If you assume that you know everything about a species, that's going to be a pretty boring career. That curiosity spurs creativity and innovation in research and gets us to think about how we might address these questions in new ways. And I think that's really exciting.
My guest today is Dr. Allison Sacerdote-Velat, Curator of Biology and Herpetology and Vice President of Conservation Research here at the Peggy Notebaert Nature Museum of the Chicago Academy of Sciences. That's a mouthful. Allison, thank you for joining me.
Thank you.
So let's talk snakes and frogs.
Yes.
What are some of the unique features of the smooth green snake?
Well, the smooth green snake is a grassland-dependent snake species. It's in the colubrid family, so it's a constricting snake. And it's only about 2 feet long, bright green, very hard to find in its grassland habitat. So that crypsis is really its best defense. It can blend in well with its environment and be largely unseen by many predators as well as many people. They feed largely on insects, so they almost have an ecological niche like a lizard, where they're eating small crickets, grasshoppers. They also eat smooth-bodied caterpillars and spiders and small day-flying moths.
Are there any particularly distinctive features to the smooth green snake?
Physically, the smooth scales, lack a keel, so they do have another congener, the rough green snake that occurs further south in Illinois. Their scales have a ridge down the center called a keel, and they actually are highly arboreal. So the rough green snakes, you would usually spotlight for them at night. They'll be up in trees and shrubs, whereas green snakes, you have to look down. Smooth green snakes, you have to look down, and that scale lacks the keel. They also have a really interesting and fascinating reproductive biology. So they are oviparous, but they have a great deal of plasticity in their incubation length. So they can either lay eggs early in the nesting season, and then those eggs will incubate for about a month, sometimes longer, or they can internally retain the eggs and kind of behaviorally incubate, so the female will spend more time in warm places, and when the eggs are about 3 to 4 days away from hatching, she'll oviposit. So you get this kind of span of incubation lengths anywhere from as short as 4 days to as long as about 44 days. The average changes year to year, but it's generally around 33 days. And there's some adaptive benefit to that for the part of their range in Canada. So they're one of very few oviparous snakes that ranges up into Manitoba and into the maritime provinces. And if you think about what an active season for a small snake would look like, it's a very short season. So it would be harder to find a thermally stable nesting site. So in that sense, holding on to the eggs and kind of bordering on ovoviviparity is advantageous.
For those who don't know, could you explain what oviparity is?
Yeah, so oviparity is where you're going to lay eggs. So many snakes are oviparous, many reptiles are oviparous, but we do have a number that are viviparous, like the pit vipers give birth to live young, and the natricine snakes, which would be water snakes, garter snakes, red belly, and brown snakes, they basically have yolk sacs with developing young in them, and they drop those, and then the young burst out of these thin membranes. So green snakes are kind of pushing that limit between oviparity and ovoviviparity. Some years they even have partially calcified eggs where you can actually see through a little transparent window and see the embryo developing. And we find those eggs survive as well.
So that isn't a birth defect, then a thinner shell
Well, it may be related to female energetic status. It might also be related to weather, because if you have a year where we have like a polar vortex, and this is when we tend to see more of them in these longer, colder winters where they have a later emergence, nesting is often starting about a week later than average, and that's when we tend to see more partially calcified eggs.
Mm-hmm. And what are the biggest threats to the smooth green snake?
So loss of grassland habitat, conversion of grassland into agriculture, heavy application of insecticide because they are insectivores. That is going to directly limit their prey base. But we find that their egg survival in the field is generally low. On average in our area, it's only about 49%. And what we see happen, particularly when we have frequent drought years, is that the eggs, they're very flexible, they're kind of leathery, but they can lose and gain water to and from the environment. So if it rains, they'll actually absorb water like a sponge and they'll kind of plump up. And then if it's dry, they start to desiccate and they dent in. When they dent in, you can still have them rehydrate if you get some rain and hatch, but sometimes they will have spinal deformities when that happens. But more often than not, those eggs will just shrivel up and they won't survive. And so that kind of abiotic influence is one of the biggest factors affecting their nest survival.
When you say grasslands, it's not only prairies, then it would be
Right.
other forms of grasslands.
Yeah, we have sedge meadows. They'll use oak savannas as well, shortgrass prairie in some parts of their range. In the northeast, they tend to use kind of coastal meadows, and there are populations in the Black Hills that use ponderosa pine forest. So that's a pretty interesting system that there, these kind of high elevation montane systems, and there are also some high elevation populations in Colorado as well.
What's the most fascinating behavior you've seen a smooth green snake exhibit in the field?
Oh, there are a few that come to mind. Right after they hatch, sometimes they'll do a defensive display where the hatchling will basically support almost its entire body, kind of up like a cobra looking posture with its mouth agape and look up. And it looks like kind of a startle response to try to ward off avian predators. So if you find one in the field, they'll kind of rear up at you, and it's very silly and muppety looking to us, but it's supposed to look very fierce to potential predators. So that is a particularly fun behavior that they will do. Another cool thing that we only learned about in 2022 is that they will actually nest within the mounds, active mounds of an aphid tending ant called the citronella ant. And so our best explanation for this is that the ant mound itself is acting kind of like an incubation chamber. It should be homeostatic. You'll have less variation in temperature, less variation in humidity, because the ants are kind of constantly moving soil around to protect their brood. And so the green snakes will lay their eggs inside the galleries of the citronella ant, and they'll hatch out. And we've been fortunate enough to be able to look into these ant mounds with an endoscope. And we can see that with the citronella ants, they basically ignore the green snakes. They'll move soil around. Sometimes they'll bury the eggs, then they'll unearth the eggs. But as the green snakes start hatching, we don't see any aggression. In fact, we see the ants kind of come up and antenate the hatchlings as they're emerging or as they're moving through the gallery, and then they just walk away. We don't see that same behavior with other ant species. So there is some interesting association there. And then after the green snakes leave the mound or go elsewhere in the galleries, the ants will bury the eggs. And my hypothesis here is that they're actually using the eggshell as a soil amendment to basically farm the vegetation that they're using for aphid tending. So I just think that's such a fascinating interaction. And since 2022, we've documented some degree of nesting interactions with green snakes and at least 14 species of ants, mainly 3 that are in the Lasius genus, but only the citronella ant is essentially, benign. The other ones will try to bite the snakes, and they just kind of flail and fling the ants off.
So this could be a mutualistic.
It could be, yeah.
But we're not sure yet.
Yes, so we've put temperature loggers into the ant mounds to try to compare the internal mound temperature to surface nests or burrow nests. They'll use crayfish burrows. They'll use structures created by other species that's generally referred to as inquiline incubation. So they'll use rodent nests. They will nest under shrew nests, which is a dangerous enterprise because shrews will eat them. Usually they are nesting under the abandoned old shrew nesting material, but every so often we will find a communal nest underneath an active shrew nest.
Which probably doesn't turn out so
not
well.
The best choice. You usually get some that still make it because they do communally nest pretty frequently. It tends to be, you'll get an early nest, like 1 or 2 females, and then there's another wave of nesting the next 5 days, and all of a sudden you'll have 60 eggs. And it just kind of continues in these waves throughout July. And what's fun is that we can, by checking on these nests every couple of days, we can track individual fate, track incubation length and survival rates, and see, is there a survival advantage to nesting early, nesting in the middle of the window, and nesting late. And that changes from year to year based on what the weather is doing.
Now, will the smooth green snake overwinter with other species?
Yes. Yeah, there was actually a 1939 herpetological note by Criddle called Snakes from an Ant Mound. And that was an inactive Formica ant mound. We have different species of Formica ants that build different height, different diameter ant mounds. And he excavated one, I believe in Kansas, and pulled short-headed garter snakes and brown snakes and red-bellied snakes and ringneck snakes out of there, as well as green snakes. And then there was a master's thesis by I believe it was Lang in 1974 in Minnesota, where he excavated the mound of Formica ulgii, and he found this vertical stratification of different species of snakes. So at the very top were garter snakes, which are the most cold tolerant, and then below that you had red-bellied snakes, and then below that you had green snakes, to the point that they were almost 3 meters down. And there was also stratification by age class within those species-specific kind of tiers. So, garter snakes are the most cold tolerant. They'll come out early. So if you get, an early warm up in March, you'll see garter snakes out basking, but you don't tend to see green snakes until generally late April in our area.
That is very interesting. Yeah, I didn't know that. So there's a stratification within. Yeah. So do we suspect that other snakes are doing this same type of thing as well?
Yes, the different species of snakes are using different structures. So historically, we had more populations of eastern Massasauga rattlesnake in the region, and they'll use crayfish burrows to get below the frost line for winter. And there are other species of snakes that will do that as well. We have Graham's crayfish snake, which is a more prairie pond, prairie wetland adapted crayfish feeding species. It's related to the queen snake, which use more fast-moving streams. Both of those feed exclusively on freshly molted crayfish, but they'll also use those structures for overwintering.
But they wouldn't overwinter together in the same...
They could.
Oh, they could.
They could. Really, our limitation is getting into the burrow and, far enough that you can... find the snakes, even if you use an endoscope crayfish burrows are not very cooperative for kind of peering into them. I have an articulating endoscope that can kind of look around, but you eventually hit, you know, some perpendicular turn, and then you just hit a wall of mud. But they will kind of aggregate with other snake species. Garter snakes and green snakes will also use crayfish burrows, and amphibians are using crayfish burrows. As well as Heinz emerald dragonfly. So crayfish are really cool because they're, you know, really a keystone species for overwintering in a lot of the herpetological community, as well as for one of our endangered dragonflies. So
come in handy.
Yeah.
Could you talk a little bit about the color changes that the smooth green snakes go through?
Sure. When they first hatch, they're kind of an olive drab, very dark green, almost gray. And after probably about 3 rounds of shedding, they start to look a bit paler, kind of a spring green. And then it's usually in that next season that we start to see this really vibrant bright green color. And I think that is adaptive because at the time of year when the green snakes are hatching, which is usually late July, August, the vegetation is starting to dry up. So as a bright green tiny snake, they would stand out in that kind of vegetative thatch. And they tend to be semi-fossorial, so they're going, you know, into the duff and into burrows. They'll crawl back into those ant galleries and just the tiniest holes you can find in the ground, and they essentially hunker down there for most of the winter, and then they'll emerge when things are starting to green up, and so they then start to match that environment.
Is it also a problem that people are capturing these snakes and keeping them or selling them?
Yeah, reptile collection, poaching is a problem. Green snakes and smooth green snakes in particular are very hard to care for in captivity and have very finicky diets. But it is a problem that puts a pressure on our wild populations, and it's not limited to green snakes. It's pretty astounding to hear what people will take from the wild and try to sell. So, you know, we generally we are not disclosing where our study sites are for that reason, to try to keep these populations safe, because every individual counts. Every snake is going to make a contribution to the reproduction, the, you know, the future persistence of that population.
Are there any laws on the books in the state of Illinois or any other state against collecting?
Yes, Illinois has a state herpetological code that is regulated by the Illinois Department of Natural Resources, and you are allowed to take up to 8 individual herps, reptiles or amphibians, but not those that are state listed, and generally not those that are in greatest conservation need. If they're threatened or endangered, you're not allowed to take them. Their scientific research permit, or, you know, captive propagation regulations that govern specific projects, but, you know, someone shouldn't go take 8 green snakes from the field. They are a declining species, so we want to protect those wild populations.
Are they a threatened species, or what is their status currently?
So they're currently a species in greatest conservation need, and they're a candidate for listing.
I want to return to the color thing, and I read somewhere about when they die, there's a color change as
yes.
well.
So they have, yeah, after death, there's a yellow pigment that breaks down very rapidly, and they appear kind of bright turquoise. If you've seen areas where herbicide's been sprayed and they mix that kind of bright turquoise stain in, so you can see where invasive plants were sprayed, it's the same color. It's just this very vibrant turquoise. And when they have an injury as well, sometimes the invasive mantis, the Tenodera sinensis, will try to kind of grab them as they're moving through the grass. We find some that have scars that are this bright turquoise as well. So if they've sustained any trauma or injury, you'll see these little spots or stripes of bright green. But other than that, they don't have any markings, any stripes or blotches or saddles on them. There's also another color morph of green snakes, referred to historically as the buff green snake. It's kind of a beige snake, and in the sun, they almost appear copper. They're kind of iridescent, very pretty. We only find those in maybe 1 or 2 sites in our area, but I know, people have found them in Wisconsin and Minnesota. So it does seem like just a recessive trait that maybe is expressing itself more in the smaller inbred populations.
Could you talk a little bit about what you're doing here at the Peggy Notebaert Nature Museum to help the green snake?
Sure. Sure. So we have a couple of different recovery program related research projects with the smooth green snake. So one of the things that we're trying to do generally is update the understanding of their current distribution, because they are so cryptic. They're easily overlooked, and unless you're actively out looking for them, you tend to miss them. However, people see them crossing trails and, just inadvertently when they're out. So we do have an iNaturalist project called the Smooth Green Snake Conservation Assessment, where if somebody sees one, they could snap a picture and upload it to that iNat project. That helps us keep the distribution records as current as possible. We can always use more central Illinois data. The range of the snake is largely concentrated in the northeastern part of the state, but historically they were distributed throughout the Grand Prairie region. So about Champaign, that's kind of the limit. There are a couple of little pocket prairies and old railroad prairies where we still find them, but there's been so much conversion to agriculture in much of that area that there are very few populations left. But keeping those records current is really critical. We are doing demographic monitoring of known populations with different agency partners. So we've worked for a very long time now, about 15 years, with Lake County Forest Preserve District, monitoring smooth green snake populations, and for about 13 years with DuPage County. We've also worked with McHenry County before, and this year we expanded quite a bit of our survey work into southern Cook County, into kind of the southwest and southeast suburbs, as well as the Calumet region. And there's some populations hanging on. But one of the challenges with green snakes is that they tend to be restricted to remnant grasslands and remnant wetlands. And we don't see kind of independent dispersal to new restoration sites if they aren't physically adjacent to those remnants, or they're not connected through like railroad ballast or a power line right of way where there's a strip of vegetation. So as part of our work to try to secure regional populations, we want redundancy. We want multiple populations in grasslands that can support the species, so that if something happens to one population, they don't blink out at the regional scale. So to that end, we do a lot of work evaluating applied conservation approaches, so reintroduction, translocation, head starting. And with the green snakes, because that egg stage is the most vulnerable, that is what we tend to focus on, promoting survival through the egg stage. So early on in the program, we did bring a few adult individuals in to try to see if captive rearing would be an option for them to increase basically our cohort size for reintroduction, but they have some challenging reproductive physiologies where they can store sperm and delay fertilization. They tend to produce smaller clutches in captive settings than they do in the field. So it was not the way to go with that program. So we also brought individuals in, like brought in eggs and hatched those out. By incubating the eggs, we can get them from the average of 49% hatching to about 98% hatching success. And that goes a long way in bolstering just the number of individuals you then have to go through overwintering. So we bring some in to the museum, and we've head started them for about 10 months. We do put them through overwintering, because with the smooth green snakes, we see compensatory growth after overwintering, and the females that have been overwintered actually catch up to females that were kept active all year within about 2 months, and they generally end up about 30% larger. So it's advantageous to give them that cue. The overwintering window is also when spermiogenesis and oogenesis happen, so it's an important physiological cue for them as well. But in recent years, we've been shifting toward focusing on just getting them through incubation so that we can work with more snakes at once. We're obviously going to be space limited with how many snakes we can house in our lab space, and you want to make sure that they're individually housed, because with any species of juvenile snakes, if you have them group housed, you can have accidental cannibalism. So we need to make sure they're eating and all of that. So you can only work with a set number of head starts. But with incubation, we can bring in hundreds of eggs, get as many of them through hatching. We measure, mark, and release them back out to their nesting site. And in the course of doing this over several years, we're seeing the number of nests increase, the clutch size increase, and just greater population growth.
But the captive breeding hasn't seemed to work so well.
No, no one had really done it, and so you try different approaches, and that's one of the really critical things, I think, in applied conservation. If you're working with understudied species, you never know what you're going to learn about them, and it may seem straightforward, like, oh, we'll just bring them in and they'll breed, and there's nuance, and I used to work at Lincoln Park Zoo, and some of my coworkers dealt with, they worked in the Population Management Center, where they're trying to basically be matchmakers for genetic diversity for breeding pairs of animals. And you can have breeding recommendations, but the animals don't necessarily want to follow it. And, this is one of the challenges of getting captive populations of wildlife to a sustainable level to support future reintroduction. You think it would be a basic thing, but they've got personalities.
Now, snakes obviously elicit a strong response in people. Most people either like them or have a strong dislike of snakes. Do you think our fear or misunderstanding of snakes has its roots in our evolutionary origin, or do you think that's something that's been introduced?
I think there is some evolutionary root to it. There have been some studies with other primates showing them snakes and seeing a reaction, and it could be a species of primate that doesn't really coexist with any other snakes, but you introduce it and they have this kind of fear response. However, I think in human culture, we also cultivate that at an early age. Snakes are, portrayed as villains in Bible stories, and snakes are portrayed as villains in kids books and movies. So Indiana Jones. Most of those were actually legless lizards. You can see the ear holes.
I'll have to rewatch
that. Yeah. There were some snakes, but they didn't have enough, so they supplemented it with legless lizards, with European legless lizards. So I think that people are taught to fear it, and I think it's something that young people are generally more open to experiencing, and that's something that we really see here at the Nature Museum, because we have our living collection, and part of the mission of the living collection is to connect our museum audience with local species that are native to Illinois. These are species of conservation concern that most people don't know occurred here historically or are still here. And, you forge that connection early. There's the saying that you'll conserve what you love, conserve what you care about. If you can get people to care about these species and make that connection at a young age, that tends to stay with them. So if you watch our public programs where we have animal feeding or a critter connection, the children are usually, oh, what is that? And it's the parents who are standing back. So that tells me it's more learned than innate.
Yeah. So let's move on to wood frogs. Fascinating little species. Let's start with their remarkable capacity to freeze and still survive.
Yes, wood frogs are one of the most cold tolerant vertebrates and certainly the most cold tolerant frog. They are the only frog that can live north of the Arctic Circle. And they do this by essentially diverting glucose from their bloodstream into internal organs as the temperature starts to drop. And this essentially acts like an antifreeze. It prevents intracellular ice crystals from forming and then damaging their internal organs. And the longer they stay frozen, they'll start breaking down muscle protein using glycogen in the same way. So they can get very skinny in the process of, overwintering, but they can stay essentially frozen for about 6 months, and about 65% of the water in their body can freeze solid, and they can survive that.
But the typical signs of life would be non-existent, right? The heart stops, there's no brain activity.
Yeah, everything just slows down to an imperceptible pace. And they just basically are in stasis. So I know there are biomedical researchers who are looking into how this works to try to develop approaches for preserving human organs for transplant so that you could freeze organs for longer windows of time. There's also, of course, people who are like looking into, human cryopreservation, so they can live forever. But I just think it's such a remarkable adaptation. And we see some other frog species that can use that same mechanism, but not to the extent and length of wood frogs and not to as cold temperature. So like I have a colleague, Kelsey Ryder, who studies Andean frogs, and they're extremely high elevation and similar cold tolerant adaptations. And there's a salamander called the Slovenian olm that also is quite cold tolerant, but wood frogs are kind of the lead.
Are there any other amphibians that are found in the Arctic Circle?
No.
The wood frog is the only one.
The only one.
So yeah, at what point in their development are they able to withstand those extreme temperatures?
So it would be in their first year. So they'll, breed in generally our area late March, early April. That's going to vary based on where you are. But they can breed when there's still ice on the pond, and they are what we call explosive breeders. So if you think of, if you're familiar with chorus frogs, which are one of the most widespread frogs in our area, their call sounds like somebody's finger on the teeth of a comb. Chorus frogs will often start calling in more open canopy sites in February and continue calling into May or June, and then you'll hear them call again in fall. Wood frogs, it's one and done. They have about at most a 2-week window of calling. Some years it's a single night, and it's just an explosion of breeding activity, and they'll lay their eggs. Those eggs will be oviposited in temporary fishless pools, so they're trying to avoid predation from fish. So then they have this race against time to try to get out of the pond as quickly as they can and develop into as large a froglet as they can before that pond dries. And then they'll move into the upland, and then they'll have to do freeze tolerance in that first winter. And they don't go into burrows. They they basically shimmy their bodies and make what's called a form, which is a little divot in the soil. And they'll just kind of hunker down and they'll have leaf litter on top of them. But that's it. So once there's snowpack, once there's ice, they're just protected by that leaf litter.
And could you talk, since there's such an explosive time for the reproduction, can you talk a little bit about how males and females court or mate? Are the females cueing for a song or vice versa?
Yeah, the males tend to arrive at the ponds first, and about 85% of them will go back to the natal pond that they came out of, unless there's a drought, and then they'll go to the next closest body of water. But the males will call, and they'll have essentially calling competitions, and their call is a two-note kind of chuck. We can think about frog calls as trills, snores, and chucks. And so wood frogs have a chuck that's like that. That's my wood frog impression. It's just two-note. And so when you hear them start to call, you'll hear one, and then you'll hear another one from another section of the pond. And as more and more males arrive, you'll start to hear a chorus, and it actually sounds like ducks quacking when they're at full chorus. And then the females are a little bit, they're overwintering a little bit further away from the pond than the males, so it will take them a little bit longer, but they are cueing in on a combination of things. They're also generally returning to the natal pond. They cue in on olfactory cues from the water. All amphibians use celestial cues in navigating to ponds, and then they are generally looking for or responding to particular aspects of the quality of the frog chorus. So generally larger males are going to call longer with a lower frequency in the case of wood frogs. It's tree frogs, they're going to call with a higher frequency and a faster call rate. There was a study from New Jersey, I think, a couple of years ago, where they had acoustic cameras monitoring different ponds, and they were finding that if I'm remembering correctly, the females were going toward ponds where there was less variance in the call frequency in the chorus. So this is where they have maybe a choice of more similarly sized mates, similarly, energized mates. And then when the females get to the pond, they have what's called scramble competition, where the males will try to wrestle each other away from the females. So amphibians have external fertilization, but a male will kind of grip onto the female, that's called amplexus, and try to get her to release the eggs into the water, and then he'll fertilize the eggs as she releases them. But what we see is other males will try to pry him off her back, and you get what we call a mating ball, where sometimes you see 3, 4 different males all trying to pull the other males away. And unfortunately, that can result in the females drowning, because whichever male is the strongest will end up kind of diving with the whole bundle of frogs to try to drown the competitor and get them away from the female. So it could be some carnage. And they're not the only amphibian that does that. We find that with toads too. It's pretty aggressive. And wood frogs will accidentally grab salamanders just in the moment.
Different species.
Yeah, it's just, oh, there's something moving in the pond. And so.
But the females, so when the females are going back to the pond, they're using olfactory senses to locate the water?
Yeah, that's tends to be more what the males will do. The females will still cue in on that as just like a navigation orientation cue, but then where they go in the pond is going to depend on the calls that they're most responding to.
But they're also navigating with the stars then too.
Yes. But if it's cloudy, then they have to use a combination of other senses. So wood frogs actually form, well, all amphibian, pond breeding amphibians will form kind of their map and compass of the site as tadpoles as they're developing. And so they're using both, celestial cues, but also the silhouette of trees against the sky to know I'm going in X direction, and then this is where I am relative to the pond. They'll come back. If they go further because maybe they need to forage, maybe there's not a good food resource, maybe it's dry and they have to get to another spot, that's when more of that olfaction will kick in, as well as vision. But the auditory cue is what kind of synchronizes that migration to a particular breeding pond.
And is it common for other amphibians and reptiles to use the celestial navigation?
Yes.
Yes.
Yes. Newts are one that also use magnetic cues as well. And they do longer overland forays in their juvenile stage, the red F stage, because they're unique in that they have this 3-part life stage. So they go from egg to aquatic larva to juvenile red F. They can spend about 3 years wandering in the upland, dispersing, and then they go to a pond and resume this kind of aquatic form with that compressed tail fin.
Do you remember the first time you saw an amphibian in the wild?
Yes.
Specifically a wood frog and a smooth green snake?
Oh, well, those I saw later. The first time I saw an amphibian in the wild was probably a red-backed salamander. My dad's very into salamanders and frogs when he was growing up, and he would take our family out to parks. And I grew up in Brooklyn, New York, but we would go to a park in Staten Island that had this nice kind of green belt, and there were lots of rocks, and we would find red-backed salamanders there, and we would find northern two-line salamanders there, and that was just such a treat. See these bright, colorful animals under logs and under rocks that look like they should be somewhere tropical. Yeah.
Is that the moment that you knew that you wanted to be a herpetologist?
That was definitely one of the moments. I knew that I wanted to work with animals in some capacity, and I kind of like everything. So I worked with mammals for a while. I worked with birds as well. But yeah, herpetology is just really what holds my interest most. I just think the adaptations that the species have are so fascinating, and they're so underappreciated, and they're in need of conservation. They're in need of spokespeople to get the public to care about them. And that's one of the things that we say here at the Nature Museum. Conservation messaging often focuses on the warm fuzzies, but we do the cold scalies.
And do we notice any physiological differences between the wood frog populations here versus the Arctic Circle?
Yes, they're going to be less cold tolerant here, just generally because they aren't exposed to those same cues, and we're having milder and milder winter. So I don't know that if you put a population, translocated a bunch of our wood frogs to Alaska, that they would particularly survive the winter. That is probably more of a local adaptation to supercooling, whereas, physiologically, they should be able to, but if they've never experienced it, I don't think they would fare very well. We also see some phenotypic differences in wood frogs here versus some of the northern and more montane populations in the Northwoods of Wisconsin. And then a former field technician of mine did her dissertation in Alberta, in the Rockies, and their wood frogs, some of them have a mid-dorsal stripe. And so not all of them, so they have striped and unstriped. The substrate in the ponds is very different. It's more rocky. There's far less vegetation in those systems. So it may be a better crypsis for them in that type of environment. Whereas here, there's a lot of detritus. There's thick leaf litter, there's pieces of bark, and they blend in so well with leaves. It's just unbelievable.
Do we know how climate change is impacting these two species already?
So for wood frogs in particular, because they're using temporary ponds, the frequency of drought that we have been experiencing is a particular hardship for them. The last 5 years we've been in drought of varying severity, and last February was the driest I had seen it in 21 years of working in these sites. I went out to the wetlands to put my call recorders up, and there was really no snowpack, really no ice, and I was essentially standing in a dry pond basin. And I went to 16 different sites, and they were all like that. I'd never seen that before. Um, it eventually refilled. The wood frogs did breed, but they didn't breed in the ponds they went to the places where the water was a little bit deeper. But even so, we ended up back in drought, and not all of these breeding ponds will hold water long enough for them. And there are some species like the prolonged breeders, like chorus frogs, that have a much shorter development time. It's generally about 96 hours to hatch. They can get out of the pond more quickly. They can reproduce again if the first round doesn't work. So there are some species that are going to be better at adapting to these kind of drought refill cycles. The explosive breeders like the wood frogs and the leopard frogs and most of our woodland salamanders like the blue spotted and tiger and spotted salamander, they're going to have the harder time. Producing eggs is a huge energetic expense, and so it's one and done. But we generally see, depending on the hydro period of the pond, some years they can make it out and they'll just be these, very chunky little froglets that rush through metamorphosis. But gosh, 2021, I think everything dried in May, and 2022, it dried early June. So that's where we start to have problems. If you have habitat fragmentation, you don't have multiple wetlands that they can go to. They're basically stuck with what is available to them. So, the general knowledge in amphibian ecology is you want a complex of wetlands of different hydro periods, but when you have permanent water, that's where you have fish. And for species that are more sensitive to that presence of predator, that's not necessarily going to be an effective refuge for them. So there are some species that are going to lose out and some that will be able to cope.
Could you talk a little bit about the work you do with the Calling Frog Survey?
Oh, sure. The Calling Frog Survey is one of 3 community science programs that we run here at the Nature Museum, and the other 2 are Illinois Butterfly Monitoring Network and Illinois Odonate Survey. And all of these are what we call longitudinal biodiversity monitoring programs. So the Calling Frog Survey dates back to about 2002, and we train anyone who's interested. They have to be at least 18 if they're working independently, or they could be younger with a parent. We train them to identify the calls of 13 regional frog species, and then we work with them to establish a survey route. And all of our survey routes for all 3 programs are on publicly accessible land that is managed as natural areas. So places where they're doing restoration, places that are remnant woodlands and prairie. And it can be urban parks as well, but we want that natural area restoration connection, because the idea is, frogs are an excellent indicator species of wetland quality as well as upland quality. They need both components to carry out their life cycle. So if you're trying to understand how effective restoration is, they're a really good benchmark. In the more diverse wetland hydro periods you have, the more amphibian species you will find in a given site. So we break this season up into 3 rounds corresponding to the phenological peaks of different suites of frogs. So we have our late winter, early spring breeders. Then we have our kind of the April guys, kind of right in the April-May corridor, mid-spring, and then we have our summer breeders in June and July. So we ask monitors to go out at least once each of these 3 rounds. If they can do more, great. They go out at night, and their route has different stopping points that correspond to different wetlands. Each stopping point needs to be at least 200 meters away so that you have some independence between points, so you're not just counting the same frogs over and over again. And we have them assign a call index to whichever calls of the particular species they hear. So it's index 1 if they just hear a few individuals with no overlap, index 2 if there's more of kind of an auditory ping pong going on and a little bit of overlap, and then index 3 if it's a full breeding chorus. So that lets us generate maps of distribution of the species throughout the region. It lets us track breeding activity over years and changes in occupancy of whether a frog species is present in a site or not. So we can see, oh, a new frog species came in following restoration, and we can see that benefit. Or we could see if a site becomes degraded that we may lose species. And then at the landscape scale, if we zoom out, we can see where there are barriers to colonization and migration, major roadways or other kind of infrastructure that may not support amphibian migration and make it difficult for a species to occupy an available restored habitat. And in that case, that may be some occasion where the land manager wants to consider a translocation to support greater amphibian diversity.
Do you have the volunteers record themselves too, or no?
We tell them they can. There are certain species that we ask them to please get an audio recording or video of because we want a voucher, and that's for generally the rare species and then the ones that are a little bit confusing in terms of the call. So if somebody hears a wood frog, we want a recording of that. If they hear a cricket frog, cricket frogs are common in some parts of the region, but they used to occur all the way up to the state line. They largely declined from the northern third of the state in the 1960s. It was a very sudden and enigmatic decline, and they are kind of slowly making their way back in places, but then we have some major roadways and major barriers to colonization. So if people are hearing them, we'd like to have that recording as our voucher.
What do you think are some life lessons that we could learn from snakes and frogs?
Oh, resilience in a challenging environment. You know, nature throws everything at frogs and snakes. They're both occupying the middle of the food web. They're both predator and prey. So they've got to hustle. They have to find their food while also staying safe, and watching their back from everything that can eat them, will try to eat them. So there's determination and absolutely resilience, especially with amphibians. You have some really long-term droughts in particular areas, and then all of a sudden, after wetland refills, you just see all of these systems teeming with life, and it's like they're coming out of the woodwork. Literally, they're crawling out of logs and out of burrows, and they've got an amazing capacity to survive really challenging changes in their environment. But dispersal is where they really end up kind of falling short a lot of times, because they can't cross roads well.
What do you think is a common misperception people have about the work that you do?
Oh, well, one thing is I think that most people expect that I have a house full of reptiles and amphibians. I don't. I like to appreciate them in nature and let them be. I like to observe them and learn from them. But yeah, I think that one of the misconceptions also is that snakes are slimy. They're not. Amphibians are slimy. um, but yeah, I think that they would be surprised at the amount of biodiversity that we have locally. I think it's a misconception that you have to go elsewhere to find, cool snakes and cool amphibians, but they were here historically. We did, through urbanization and land conversion, we eliminated a lot of species and eliminated populations. We're working to try to reestablish our historic biodiversity. And it's really important to look around and learn about what was here, what is here, and what we're trying to conserve in the Chicago region, because we have some really amazing habitats.
And well, to dovetail on your comment about you don't have a house full of, what is your stance on private ownership of?
Yeah, I mean, I think there are a lot of important contributions that come from the husbandry hobby. We've learned a lot about captive rearing and environmental needs. It just has to be done with permits, and we have to make sure that we're not impacting wild populations to the detriment of nature. It's a delicate balance, really. People like to appreciate them and like to take care of them, but taking them wholly out of their environment is a disservice to the ecosystem. Additionally, we have to be really careful about pathogen transmission. There have been a lot of detrimental pathogens introduced through pet trade. So if you're going to keep something, don't turn it loose into the environment when you get bored of it. You have to consider the life history of the species, because people turning red-eared sliders loose introduces pathogens into our native wetlands, and people introducing newts from Asia into Europe introduced salamander fungal disease. And now this is something that we have to do surveillance for and biosecurity for and conservation work to address.
Was the chytrid fungus that wasn't introduced via that, was it?
So we have a historic strain of chytrid fungus in Illinois. There was a study in 2015 by Talley et al. That went through museum specimens, and they were able to isolate chytrid in southern leopard frog species from 1899, I believe. So it's been around forever, but there are, well, forever for a time that we're able to reflect on through the use of natural history collections. But there are different strains that have different pathogenicities and different capacities to harm populations. So there's like the pan-global strain, which is a more aggressive strain that is causing mass die-offs in much of Central America and South America and Australia and New Zealand. It's not the same strain that we have here, but with movement of animals around the globe, that's something that could be very easily introduced to our ecosystem.
And what do you think is the most important quality that a biologist should have?
Curiosity. I think you should never stop asking questions, and you should never stop trying to understand behaviors or phenomena that you see. If you assume that you know everything about a species, that's going to be a pretty boring career. I've worked for 15 years now on smooth green snakes as a focus, and for 21 years on wood frogs, and there's still so much, so much that I don't know, that especially about overwintering ecology. We can't shrink ourselves down and get into these burrows and follow them as long as we might like to. So that curiosity spurs creativity and innovation in research and gets us to think about how we might address these questions in new ways. And I think that's really exciting.
Has there been someone that has changed the trajectory of your life
Oh,
as
sure.
a scientist?
Um, oh, lots of people. Um, yeah, I would say one of the people that I've worked with who really showed me what was possible in urban ecology was Robert Cook. And he's a biologist. He's now retired, but he's a biologist with National Park Service. And he had done work in American Samoa, but was based in New York for a long time, and then Cape Cod. And he undertook an extremely ambitious herpetological repatriation effort in New York City. Of all places, you don't think of that necessarily as a hotbed of biodiversity, but he did work in Jamaica Bay, Rockaway, and Sandy Hook, all these sites around New York Harbor. And I had the good fortune to work on a project with him that was I was a field technician for in Sandy Hook, New Jersey, and it was 10 years after he had reintroduced Fowler's toads and hognose snakes to this barrier spit off the coast of New Jersey. And so we were doing work to inventory all of the reptiles and amphibians in different national parks as part of an inventory and monitoring effort. But in those sites, we're specifically trying to see, did his reintroductions work? What's still here? What needs more conservation? And we had different teams working in Queens and in Brooklyn and Long Island, and just the amazing amount of species that he was able to work with, like racers and box turtles and milk snakes, and it just bring it back to what it was. And I found that incredibly inspiring. One of the things we try to do is, you want to keep the common species common, but we also, the Academy's focus on the biodiversity of the region historically informs us of what was and tells us what could be as we progress through restoration. He also told me trash is habitat, and I filed that away. And
Do you agree with that?
yes and no. It depends on the kind of trash, but I will say working in Calumet this season has been fantastic. And these sites have been through the ringer. They've been contaminated slag from the steel industry. They've been capped and they're undergoing restoration. But we find so many cool things there. And there was a particular site where there was a license plate that my technician and I would flip every time we went to the site. And I said, one day we're going to get a green snake under here. One day we're going to get a green snake. Of course, um, just a few weeks ago, tail end of the season, I had a meeting, so I had to be in the office, and my technician texted me a picture of the license plate with the green snake. I'm like,
There we go.
can't believe I missed it. It's the epitome of urban herpetology.
Yeah. What would you say is your most memorable experience in the field?
Hmm. The first time I saw a Gila monster, which was when I was working in Utah. Um, I was on a desert tortoise survey crew, and we were in a particular site that our boss had told us. So yeah, this might be a good chance for you to see Gila monsters. And I walked around a bush, and there's just sagebrush. There's not a lot of cover, and just stopped in my tracks to see a Gila monster in the wild was just such a thrill.
Yeah.
And then I got to help with a radio telemetry project with them um, a couple of years later, and they're just incredibly charismatic.
What do you think is the biggest threat to reptiles and amphibians that people might be unaware of?
Generally, uh, I think pathogens are one of the ones that we need to build awareness about. I think most people are aware that habitat loss is the biggest driver of extinction and biggest loss of cause of loss of biodiversity. But there are so many pathogens that we are dealing with, whether it's snake fungal disease, which we have in the region, whether it's chytrid or salamander fungus or ranavirus. There are some devastating diseases that have the capacity to wipe out populations very quickly. And that's, I think, why it's critically important to conserve as many of the populations as we can on the landscape, because you never know what's going to happen in one particular site. And we see resilience to some of these pathogens, but you know, when COVID hit us, we, you know, those first few months, it hit us hard, and it's no different with pathogens for wildlife.
How has your work studying reptiles and amphibians impacted your own humanity?
I think it's given me a greater appreciation of, you know, how important it is to be able to adapt to change. You know, we have changing environments, we have changing threats, and we are not going to be resilient as a society unless we are willing to make changes in the way that we do things and the way we approach problems. What has worked for us in the past may not continue to work for us in the future. And you see that with wildlife all the time. You see there's new competitors, new challenges, resource shortages. And so you have to think differently about how we will deal with problems as they arise, and not assume that we're in this stasis where everything will be as it was.
What are some of your future hopes and goals, both with your research and your career?
Well, I have to say I'm in my dream job. So, you know, I get to do the work I want to do, which is, you know, conserving species I care about and partnering with agencies that are doing the boots on the ground work to restore the habitats and conserve the habitats. And what I would like to see is probably greater movement toward landscape scale conservation and kind of broader projects of broader scope. Oftentimes our funding is very tied to a particular region, a particular site, or a particular county. And when we want to work with a species, you know, kind of at that zoomed out range wide scale, that's something that's going to take more collaboration. So one thing I've been working on is reaching out to other partners in the Midwest and the Northeast who may have green snakes as a listed species in their state, or it's on their radar as a species in greatest conservation need. And we've been talking more, trying to pool our knowledge and understand how the species is dealing with some of those same kind of life history challenges, like nesting or, you know, dispersal in different environments throughout their range. I think we'll get the best sense of the potential for recovery if we have more of a range-wide assessment. And I think that that's the case for most species of reptiles and amphibians that we're working with.
Great. Well, Allison, it's been a pleasure.
Oh, thank you. It's been
Thank you
great
for the
talking with
time.
you.